Hey guys, today we're going to talk about DNA a little bit and kind of the background of it and how scientists figured out that it is a double helix, it has four bases, and where that came about. So, if you look at the slide and on the notes, two men by the name of Watson and Crick put together a model of the structure of DNA, which kind of showed how it could serve as the genetic material. And we're going to talk about that in another set of notes. But they help determine that DNA is what we call a double helix, which is two strands twisted around each other. You can kind of think of it as a spiral staircase, is how a lot of people like to think of it, or Twizzlers, something like that, something where two things are wrapping around each other. If you look at the picture, this is exactly what I'm talking about. So in a minute, we will talk about the different base pairs, but you can see that there are four base pairs in DNA. You have adenine, thymine, guanine, and cytosine. And most of the time, we will use those as abbreviations. We will call them A, T, G, and C. I will never expect you to learn how to spell those, but you do need to know that they are there, and that is a huge part of DNA. You also, if you look at the picture, can see that the blue parts on the outside are what we call the sugar phosphate backbone. And again, we're going to talk more about the different parts of the DNA here in a minute, but this kind of just gives you a preview when I say double helix of what I'm talking about. Each strand is made of what we call a nucleotide. And the nucleotide is just a subunit that makes up DNA, and it's made up of three parts. You have a phosphate group. So if you look at the picture, you have a phosphate group here. Then you have some type of 5-carbon sugar, and then what we call a nitrogenous base. And if you look at that word, you can see the word nitrogen in there. So any nucleotide is made up of those three parts. Now, the base differs, and you can see in this picture that they are using adenine, but based on the last slide, you could also use thymine, guanine, or cytosine as well. The 5-carbon sugar in DNA is what we call deoxyribose. The full name of DNA is deoxyribonucleic acid. Again, I'll never expect you to know that or how to spell it, but that is what DNA actually stands for. Okay, so you have a sugar molecule and a phosphate group, and this could be the same for each nucleotide, which is that subunit we just talked about. The nitrogen bases, like I previously said, can differ. It can be A, T, G, or C. And if you look at the picture down here, you can see what the structures of each base is. I don't expect you to draw those. I will tell you in high school, I'm sure you will have to do something with those structures. But in our situation, you just need to know that they exist. And then again, here's that double helix picture we were talking about earlier. A and G, adenine and guanine, are what we call purines. And basically what that means is they are bases that are made of two rings of carbon and nitrogen. If you look at the picture down here, this is A, this is G, and you can see they have two separate rings on each of them. Thymine and cytosine, however, are what we call pyrimidines, and they are made up of just one ring of carbon and nitrogen, and you can see that down here below. You can clearly see the difference between the one and the two. There's also a pyrimidine called uracil. We will talk about that when we get into RNA. Uracil is not a part of DNA, so we're not going to talk about it at all right now, but we will visit that later on down the road. Okay, so kind of the background about how they figured out the bases, how they figured out how they're purines, pyrimidines, and what DNA is made of. There's a scientist by the name of Chargaff, and he conducted an experiment, and his data ended up showing that for each organism he studied, the amount of adenine always equaled the amount of thymine. And then on the opposite end of that, the amount of guanine always equaled the amount of cytosine. So A equals T, G equals C. Okay, now because of this, A always pairs with T and G always pairs with C. We'll see that in a minute. But that does not necessarily mean that it's the same for every organism he studied. So for instance, in humans, we have a different amount of AT G and C, as opposed to a dog or a whale, a fly, something like that. So it's not the same across the board, but for each individual organism he studied, they always matched up. Chargaff's data, and I know we've talked about this in class before, that scientists kind of play off of each other. So his data became clear when two other scientists, Wilkins and Franklin, came around and they conducted an experiment where they used what we call X-ray diffraction. And that typically is used to study the structure of molecules. So these two scientists came along and they took an X-ray beam of electrons. And what X-ray diffraction is, you will shoot a beam of electrons at some type of structure or molecule. In this case, they use DNA. And the electrons will bounce off of that molecule and kind of splatter onto a sheet. And then they can use a flashlight and they can look at the structure and determine what that particular thing looks like. So in this example, talking about DNA, this is what the electrons look like when they got shot onto that piece of paper. And you can see, well, it's hard to see, but it kind of looks like a double helix. These are all representing bases as well. To us, it probably doesn't make much sense. To scientists at the time, this was a really big deal, and it really showed them a lot about the structure of DNA. We always joke around in class that this little guy in the middle kind of looks like an Indy car. That's usually all I see when I look at it. But you can kind of see if you're standing down a spiral staircase that these guys might be twisting around each other. Okay. And then two more scientists, Watson and Crick, came along and they used this information. They used Chargaff's data, knowing that A equals T, G equals C. And they also used Wilkins and Franklin's photographs to build a tenon wire model of what DNA looks like. So I keep throwing out the term spiral staircase because you will just always hear that. I mean, that's always what it looks like. So if you look at this picture, you can see, you can kind of see, I know, depending where you're looking, but here's a ring here, and they are just showing you A, T, G, and C paired together, wrapped around. Again, to us looking at it, it looks cool. You probably can't really dissect it a lot from a picture, but this was a really big deal to show the whole structure of DNA, the sugar phosphate backbone, so on and so forth. So in Watson and Crick's model, a purine on one strand of DNA is always paired with a pyrimidine on the other. So like we said earlier, A is always paired with T, G is always paired with C. Okay, and Chargaff showed us that as well. The structure and size of the nitrogen bases allows for only this two paired combinations. You can kind of think of DNA as a puzzle to where, and I'll show you again here in a minute in a picture, but the shape of A can only fit into the shape of T, or the shape of G can only fit into the shape of C. Later down the road, we'll talk that there's kind of like a proofreading DNA polymerase that comes along to make sure that that's what happens, that those are the things that are paired together. And the base pairing rules, again, are supported by Chargaff's observations because that's what he found. A always equals T and G equals C. And this is what I was referring to when I said it's kind of like a puzzle. You would not be able to fit A and C together, and you also wouldn't be able to fit A and G together because it wouldn't fit. So these are the only combinations that are allowed when DNA pairs together. Okay, so the strictness of base pairing results in two strands that contains complementary base pairs. I'm going to X out of this here to where I can edit it. For example, if I would give you the strand TGC, AATC and ask you, hey, that's one strand of DNA, give me the opposite strand. You always just have to remember A goes with T and G goes with C. So if we look at this, the opposite strand would be A, C, G, T, T, A, G. Okay, and it's as simple as that. Later when we introduce RNA, it kind of gets a little bit more difficult because different base pairs switch out. But for now, it's as simple as that. Every time you see a T, it goes with A, G goes with C, and vice versa. Okay. We will definitely go over questions you have in class tomorrow. For now, make sure you go back to Google Classroom and follow the next step of what you should be doing in your assignments.